Today we're going to talk about the 2017 corn silage trials. That's a collaboration between Cornell University and the University of Vermont, where we evaluate corn silage hybrids submitted by seed companies across the two states at various field locations. I'm Joe Lawrence, a dairy forage system specialist with the Cornell ProDairy program. So as I said, this is collaboration between Cornell and University of Vermont. There are a lot of great people involved in making these trials happen every year, as well as a lot of support from our funders, Farm Viability Institute, Northern New York Ag Development Program, dairy producers across the region, the seed corn industry, and Cornell Cooperative Extension. These were the participants we had in the 2017 program. So each company has the option to participate in the program, and they can select hybrids of their choosing to be submitted into the program. And we really value their participation, because without that, the project wouldn't happen. So just to jump right into some of the forage quality side of it here, because when we're evaluating corn silage, it's more than just looking at yield. We're really interested in the forage quality aspects of the silage, and this is a slide from Dr. Randy Shaver at Wisconsin, but I think it does a nice job summarizing some of the main indicators of forage quality for dairy animals. And so the down arrows indicate when lower numbers are better, so NDF lignin, UNDF 240, which we'll talk more about. You want to see a lower number where NDFD 30, which is digestibility, TT NDFD is total tract, NDF digestibility, starch, and number calculations like milk per ton, you're looking for higher numbers. So to go back a little bit, so NDF is neutral detergent fiber. It's been a key measurement in forages for many decades, and several years ago they started looking at the actual digestibility of that fiber instead of just the fiber level itself. And this is some data from 2005 just showing that when we measure NDF and lignin, we can have different samples with very similar contents, but then when we actually look at the digestibility of that fiber at 30 hours, there's a wide range. So same NDF, a very wide range in the digestibility of that NDF. So that was a very important concept that has came about and was intertwined into how rations are developed for dairy animals. And then more recently, the term of UNDF, or undigested fiber, has gained more interest, and we measure that at 3 different time points. The diamonds here reflect 30, 120, and 240 hours. You can see that there's a pretty rapid digestion in the first 30 hours. That's that energy value, the fermentability of that fiber, and then going out to 240 hours has really been shown to be a good predictor of gut fill or potential dry matter intake of that forage. And the other thing we see here is 2 different hybrids from our trials that just gives you an example of how that pool of undigested fiber after 240 hours can vary between hybrids. Here you see hybrid A with a significantly larger pool of UNDF at the end of that 240 hours based on the colored portion at the bottom of the graph. So we looked at how forages with the same NDF can have different NDF digestibilities at 30 hours. Now this is looking at NDF digestibility at 30 hours versus that UNDF at 240 hours. And we see the same thing here, that if we say pick the NDF digestibility of 60, we could have more than a 10-point range in UNDF at that same NDF digestibility. And that current work going on and feedback from the industry suggests that 10-point difference in UNDF is definitely meaningful to the animals in terms of how much they can eat and their intake potential. You'll also notice here that the green crosses from 2017. There's a little tighter relationship than there was in 2016, but I don't think we can count on that every year. I think the weather plays a factor there, and so year in, year out, we may have some years that have a tighter relationship than others, but overall, it's important to look at UNDF 240 because it's offering us information that the NDF D30 may not be. 2017 trials, we had 73 hybrids entered at locations across New York and Vermont. We broke it down into two maturity groups. 80 to 95 day were planted at 3 locations. 96 to 110 day were planted at 3 locations. And the blue circles indicate our 2016 sites. So 2016 was our first year of reestablishing the program. And we had 2 locations here in New York, and we'll refer back to some of that data. So just a reminder of the year, not that most people want to be reminded of the 2017 weather, but in general, very wet weather across all of our locations, as it was across much of the Northeast. Growing degree days below normal growing degree days for the growing season. So we had a wet, cool growing season. Conversely, 2016 almost couldn't have been more opposite in terms of being quite dry, but well above average in growing degree days. So what are the impacts here? There's been some of our work, some other work by Dairy One Forage Lab. The study they've done has indicated that rainfall, both monthly and seasonal, as you increase rainfall, we see a decrease in fiber digestibility. Also, soil drainage class seems to, when you have a poor drained soil, we seem to see lower fiber digestibility. So those are important things that we're gonna kind of look at through here. Starch content, I think, definitely is just more driven by the season, the crop's ability to mature. In 2017, we had some late planted corn in a cool growing season, so some farms were forced to harvest some immature corn silage. That'll affect starch as well as the whole plant dry matter at harvest. If we compare that UNDF 240 value between the 2 growing seasons, again, a lower number is better here. So a lower number means less of that undigested fiber. And so we see the blue bars from 2016 indicating that, on average, we were shifted more towards the lower end of the range, where we moved to 2017 and we see a higher percentage of the samples from our trials shifted to the higher end. And that's definitely being seen by dairy producers who are feeding those 2017 corn silages, and was a noticeable shift from the 2016 to the 2017 crop. Just to look at the crop yield plotted against the predicted milk yield. 2016, it was kind of interesting. We had very distinctive yield differences at the 2 locations. The Madrid site was below average in rainfall, but seemed to have enough rainfall to get by and had some pretty good yields, where Aurora was quite drought stressed throughout the season, and the yield suffered as a result of that. And so we really see, no overlap at all in the yield data between the 2 locations. And this is the exact same hybrids planted at both locations. If we look at that predicted milk yield as an indicator of quality of these forages, then we see that there's a little bit of overlap there. I've excluded the blue dot all the way to the right because that's a BMR hybrid. So I just looked at the conventional hybrids and just see, kind of a little bit of overlap. But in general, you saw where the drought stress corn at Aurora yielded less, but was much more digestible, and therefore had a higher predicted milk producing potential. If we jump to 2017, I have this broken out by the two different maturity ranges. So the 3 sites where I had the shorter season corn, you can see that there's 2 sites were pretty similar in yield, where the Willsboro site was slightly lower in yield. And there was kind of a range across the sites for that predicted milk yield. And interesting that again, that the Alberg, Vermont site, with some of the highest yielding stuff, had a pretty good growing season, but a lot of rain on heavier soils, and had lower digestibilities, which translated to lower milk yields. The Albion site, on the other hand, still above average rainfall, but more moderate, and better drained soil. Overall, a pretty good growing season there, and we see much higher predicted milk yields, in addition to some pretty competitive yields. Again, these were the same 23 hybrids planted at all 3 of these locations. Then if we shift over to the longer season corns, we see that there was some spread in yield across the sites, but really the fiber digestibility and the corresponding predicted milk yields, really a lot of overlap there. So just interesting to contrast the 2 seasons and the type of growing conditions each location experienced, and how that's impacting not only the yield but the quality. So as I started out saying, this really, when we look at corn silage, it's not just about yield, it's about quality. And I like to say that this is an evaluation of hybrids, not a hybrid competition. It's not a competition for the highest yielder. It's an evaluation of how these hybrids perform relative to one another. How consistent how do they perform within a location compared to their peers, as well as how consistent do they perform across locations, because each location has its unique set of growing conditions. So if you have consistent performance across locations, you know that that hybrid isn't as susceptible to some of the unique growing conditions that an individual site might have. So using this data to figure out what hybrids perform best on your farm, on your soil types, the length of growing season you have, is very important, versus just picking the highest yielding hybrid. And then what factors impact predicted milk yield the most? Well, we see that digestible fiber is a strong indicator of predicted milk yield. So we know that that's a case, and we've seen from some of the data that growing season affects that digestible fiber. And then what affects digestible fiber, right? As I just said, it's soil type, weather, yield. We see all of those factors playing in here. So at the end of the day, what factors match your feeding program and your needs the most? Is it yield? Do you have acreage constraints and you really need yield? Do you need starch? Is it the digestible fiber? And what other forages are being fed? Do you have some really digestible haelages that you're feeding with the corn silage? Or do you need that higher digestibility from the corn silage? All those factors are going to go into identifying hybrids that not only grow the best on your farm, but work the best in your feeding program. So as we get into the data here, and I'm not going to share all the data today, at the end there's a link to our full report which you can look at. I'm just going to kind of review how to use the data. So as we compare data, kind of a trick question here: is 26 tons a good yield? We really can't answer that question unless we look at what the plot mean was. So what are we comparing that 26 tons to? If the average of that location was 21 tons, then yes, 26 tons is a good yield. However, if the average at that location is 31 tons, then that 26 ton yield doesn't look as good. So that's why we want to really pay attention to what the plot mean is, and how we use that data, and how a hybrid performed relative to everything else at its location. And just to look at this again, I shared that slide earlier from Randy Shaver, and looking at thousands of forage samples across 4 different laboratories, he found average UNDF and NDF D values. And so I just looked at our numbers for the last 2 years and looked at how many of our samples fell within those average ranges. And you can see here it's a fairly small amount. If we put it on a percentage basis, it's very small for UNDF, under 20% in 2016, under 10% in 2017, and then a little higher for the NDF digestibility at 30 hours, but still relatively fewer samples. And with that, I just bring out that's why we really want to compare these numbers to the average at that location and not have a certain number in our head and not say a UNDF of 24 is a good number, because it's totally driven by the year and the growing conditions. So we want to understand that the value is how it compares to that growing season, not how it compares to the average of several thousand samples across many years and locations. So using our public data for the trials as a comparison on your farm, looking at those average digestibility numbers. So if one of our locations has a similar growing season to where you are, you can look across this table and see what the average yield, the average starch, the average UNDF 240, whatever numbers are important to you. You can see what the averages were at those sites, and look at your own hybrids that were grown on the farm to see that how your crop performed relative. And I think that's an important way we can use the data that comes out of our public trials. And just another thing here is to watch for the units. So with something like UNDF 240, it can be reported on a percent NDF or a percent dry matter basis. So it's fine to use either depending on what you're talking about, but just to be aware that that it makes those numbers look quite different, and you want to watch what the units are. So we like to use these quartile plots to plot the performance of the hybrids, and in this case we're plotting the crop yield versus the predicted milk yield. And so this is just a graphic of how to read these quartile plots. So if you're in quartile 2 in the green there, the upper right corner, then you're above the average at that location in both yield and quality. So the cross in the middle is the average for that plot. So that upper right corner puts you above average in yield and quality, where quadrant 4 in the lower right corner would be above average in predicted milk yield, but below average in crop yield. Quadrant 1 is above average in crop yield, but below average in predicted milk yield, and quadrant 3 would be below average in both. So that's how we look at these plots, and I think it's useful to look at where hybrids land in the plots. And as I said, you can find these for every one of our locations. You can find these in our report. So this is just an example from our the long season corn that was planted in the Alburg, Vermont site, where we had 49 hybrids planted at this location. And there you can see the scatter here. Again, that top right corner would be above average yield in milk. And another thing to point out here is the axis are on percent of plot mean. So the dark lines that go across at 100% means that was the plot average at 100%. So if you go up on the crop yield to 105%, what you're looking at is if you follow that line across to say hybrid number 28, is you're looking at a hybrid that yielded approximately 5% better than the plot mean. And that, again, that's more important in this case than the actual plot, what the actual plot mean was, because it's really looking at a percent above or below that average. Now, if you want to know what the plot mean was, if you look at the lower right corner, then it tells you that the crop yield was, the average was 28.4 tons to the acre. And we'll see, so we could put that at the 100% line. Similarly with the predicted milk yield, that's at 82.8 pounds per day. And so we could put that at the 100% mark for the predicted milk yield at 82.8 pounds per day. So that can give you a reference point of what your average is. The other thing that's really important here is these are replicated trials, so we're able to calculate what the least significant difference, the LSD, is in terms of what's that difference that's actually significant, where versus a difference between the hybrids that may be numerically different but is not significant. In this case, in the lower left corner, you'll see lines representing the least significant difference. And so I liken this to using an atlas or a map with a scale at the bottom. If you were to take a piece of paper, the length of those lines in the bottom, and put it up on the graph, it would give you an idea of whether that hybrid was statistically better or worse than its neighbor, or if it fell within that range. For the purpose of this, I've actually put an example together. So if we draw that line both above and below the hybrid, and if we take that, and I just picked a hybrid that was kind of in the middle of the chart, so that gives us an idea for hybrid number 9 of anything that's statistically the same or different. So basically, on a yield basis, any hybrid that falls within this shaded zone statistically has the same yield as hybrid number 9, where anything outside that shaded zone would be statistically different. If we do the same thing for the predicted milk yields and shade that, it means that in reality at this location, a good share of the hybrids had statistically the same predicted milk yield, even though there were numerical differences, where there are some on the high and low end that would be statistically different than that hybrid number 9. So this just gives you an idea of how to use this information and interpret those LSD values. And this is just to make you aware that in some cases we report different things on these axes. So in this case, we're plotting crop yield against UNDF 240, and as I said earlier, lower UNDF 240 numbers are better. So in this case, it shifts. So instead of looking at stuff in the top right corner as being above average in both, it's actually now the top left corner, or quadrant 1, that has the above average crop yield and digestibility. So I just bring that up as an important factor to make sure you know what's on the axis and what quadrants you want to be looking at as you look at some of these. And we have some collaborative efforts going on with PDMP, Penn State corn silage trials, and if you look at their report, you'll notice that they have some plots like this with UNDF 240. So again, you just want to make sure you know what you're looking at. Switching gears a little bit to tracking performance on your farm, the Nutrient Management Spear program has done some really interesting work looking at yields across fields on a farm and the standard deviation, and asking the question of what fields are most consistent. So again, this is another one of those quartile plots, but in this case, it's looking at yield versus the variability in that yield. So here we would look at the green area, the shaded green area, as these are high yielding fields, and they consistently yield high because there's a low standard deviation or coefficient of variation depending on which one you use. And then in the blue shaded area, those are also high yielding field, but there's much more variability from year to year at those fields. In the yellow, you would have lower yields but a lot of variability, and in the red, you would have consistently lower yields. So that's an interesting way to evaluate your fields and look at yield. I would suggest that we could do the same thing in terms of looking at forage quality and hybrid performance. That we could build our own database on each farm by tracking not only yield but quality from year to year, and do that both take samples by field as well as by hybrid to understand what hybrids perform the best on your farm and what fields tend to have characteristics that produce higher quality, better digestibility forages on those fields. So obviously tracking yield is an important aspect and something you can do, but also taking some samples at harvest when you know what field and what hybrid those samples came from, and tracking digestibility that way, I think it could be a valuable tool and a valuable piece of data to build on your farm. Now getting into some other areas of the trials. So we try to do other work with our corn silage trials beyond the standard yield and quality reporting. And here we're looking at a study we had funded this year from New York Corn Growers Association and the Northern New York Ag Development Program to look at western bean cutworm damage and mycotoxin. So we assessed plots at 2 of our locations for ear damage prior to harvest, and then at harvest we collected samples for mycotoxin screening. And if we look at that, the reason we did this is because western bean cutworm is becoming more of an issue in New York State. Numbers have been increasing, as you can see here from the trap counts from the New York State IPM program. And these pests have potentially can do a lot of damage to the ear of the plant. And for silage, one of the concerns with that ear damage is it a pathway to introduce molds and mycotoxins from the damage created. So we saw that we did have moths trapped at both locations, a higher amount in the North Country at Madrid. The same 49 hybrids were planted at both locations. However, there was over twice as many hybrids at Madrid. 32 out of the 49 hybrids had ear damage from western bean cutworm, where at Aurora only 14 of the 49 hybrids had damage. However, when we look at the mycotoxin screening, we see that basically the same number of hybrids screened positive for some level of mycotoxins. And to take that a step further, at the Aurora site, only 4 of the hybrids with damage had screened positive for mycotoxins. 13 of the hybrids that screened positive for mycotoxins actually had no western bean cutworm damage to the ears. So this would suggest that there was no clear link between the western bean cutworm damage and the mycotoxin development. However, this was a very wet year. In talking to Dr. Gary Bergstrom, he indicated that silk channel infections could have been a significant player in ear infections this year, and so that insect injury later in the season may not have been relevant. And so we would like to monitor this through additional growing seasons to see if we get a different pattern in seasons that are less conducive to that silk channel infection. And we will be repeating this study in 2018. So we will have a different growing season to look at. Another interesting observation we made was some significant eye spot pressure at our Madrid location. And here are the 49 hybrids that were planted in Madrid. And these ratings are averaged across the 3 replicates, looking at leaf disease. So a 1 would mean virtually no disease, a 5 would mean heavily diseased. And you know, these hybrids are all planted side by side in small plots in a field. So the fact that we see very different readings, leaf disease ratings between the hybrids suggests that there's a strong level of resistance to eye spot in some hybrids, where other hybrids are much more susceptible to the eye spot. And that this is just an observational thing to highlight that, looking at disease ratings in your corn guides to see what hybrids have different levels of resistance, it does play out in the field in certain growing seasons, as we saw in 2017. Another question that we often get asked is there a difference in performance between the conventional hybrids and the genetically engineered hybrids, ones with either a glyphosate tolerance or a stacked trait package? So I just pulled our data together from our trials here, looking at yield across the hybrids, and we really see no difference there. And what's important to recognize here is that these hybrids are planted in plots where we minimize the stress. And one thing that our genetically engineered hybrids do is help mitigate some stresses, either from pest pressure or from weather events. So this is showing that under good growing conditions, where there were no additional outside stressors, well, beyond the wet growing season we had, but that the performance is very similar. And this is important to think about, because we have some very good conventional hybrids out there that can be used. They have under the right conditions, they have a equivalent yield potential. However, they are more susceptible to some of these incidences of stress, like pest and weather. So really what our genetic engineering technology has helped us to do is close the gap between yield potential and actual yield, as we're eliminating some of the stresses on the plant. So just wanted to point that out as we wrap up here. So here's a link to our trial results, where you can also find past year results as well as 2017's for New York. And then the Penn State trial results. As I mentioned, we've had some really great collaborations with the group there on their silage testing program. And we're continuing to build those relationships, as well as building the relationship with Vermont and other areas of the Northeast. And we'll continue to work on that and to advance the evaluation of corn silages. And just to wrap up, we're excited to be getting going on another year here, and we had good participation for our 2018 trials. I'd like to acknowledge everyone that's entered the trials this year, and we're looking forward to another good season and some interesting information to come from it. Thanks for your time, and here's my contact information in case you want to contact me with any follow-up questions.